Hey you guys, welcome to 18.2. We're talking about regulation at transcription level. So let's get started. So regulation occurs during transcription in a couple of different ways. There are going to be general transcription factors, which is what we've been talking about. The transcription factors that attach on at the promoter region and allow for the RNA polymerase or creates a greater affinity for their RNA polymerase to attach onto the DNA. We also have specific transcription factors. And so these are going to be used for cells that have a more specific function. And they're going to allow for a higher rate of expression. So genes that depend only on general transcription factors tend to be expressed kind of slow and steady, so at a low rate. And so genes that need to be expressed more often or at specific times are going to have these specific transcription factors. They're going to help with the sort of specialization or the highly specialized cells. And these specific transcription factors of highly specialized cells are going to bind to a site that is far away from the operon. It can be thousands of nucleotides away. Let's take a peek at a picture. So here is our promoter region, the top box, and our gene. So right here is our operon that we've been talking about. And this is where our general transcription factors are going to stay or attach on. And now we're talking about over here, this is where our specific transcription factors are going to bind. So it is an enhancer region, and it's thousands of nucleotides away, potentially. And here, what's going to attach on are activators, and they attach on to the distal control elements. So within the enhancer region, there are segments of DNA called distal control elements. And what binds onto these are activators. And when these activators bind on to the distal control elements, what's going to happen is it's going to take this segment of DNA that is far away from the operon, and it's going to bring it, sort of loop it around. If you can see here, here's the operon. Here is the enhancer region, and you can see that it's looping around, and it's going to attach onto, right here we have a group of mediator proteins. So this is going to mediate or sort of facilitate the interaction between this enhancer region and the operon itself. So this mediator protein is going to attach onto or bind to the general transcription factor. So it's a protein-protein interaction. These activators that we were talking about right here that attach onto the distal control elements, if we look very closely at them, there's two domains. And they're under research right now, but it is understood that there are two domains. We have one domain right down here. This is the DNA binding domain. And this is the part of the protein that's going to bind to the DNA. And right here we have the activation domain. And this is going to be the part of the protein that's going to interact with the other proteins. So right here, or the mediator proteins. So here is our activator, and it is going to be bound, and then it's going to interact right here with the mediator proteins at the activation domains. So when they bind to the control elements, right here, the activators bind to the control elements, that's going to make the DNA loop around and interact with this group of mediator proteins. And this will allow for and then the mediator proteins are going to interact with the general transcription factors, and that's going to initiate transcription. So on average, an enhancer, the enhancer region right here, which is thousands of nucleotides away and has control elements, is going to have about 10 control elements, but of course it's an average, so some are going to have more and some are going to have less. And so each one of these control elements are going to bind with one or two activators. So here are our activators. Many genes require enhancers or these control elements. And one would imagine that you'd have one activator for each control element and one control element for each operon. But it's not really like that. It is, think once again that we have one message and we are using different means to express it in different ways. So we have the same, actually over and over again, we use the same control elements. So we have the same nucleotide sequences that's used over and over again. So we can see here we have a yellow, a gray, and a red representing. And here, down here, for a different cell, we have the gray again, and then a brown and a pink. And so it's going to be, these control elements and activator proteins are going to be used in different combinations, and they're going to be used over and over again. Sort of like the alphabet. We have the same letters that we use, and we just put them in different orders to make different words. And so it is also these activator proteins. We have a finite number of activator proteins, which can bind to one or two different control elements. And when these are around, they're going to signal the expression of that gene. And if they're not available, then that gene cannot be expressed. And so this is going to indicate when a liver cell should be turning on the I'm a liver cell gene. It's when those specific activators are present. And that's going to depend on where the cell is and the environmental factors. So each combination of control elements can activate transcription only when those specific activator proteins are present. So this is, like I said, what makes a liver cell, and this is what makes a lens cell. So we can see that we have the same control units, but they're just in a different order, or the control elements, they're just in a different order. And the types of activator proteins that are, or activators that are available to bind is going to indicate when and what will be expressed. So the environment is going to play a role in what activators are available. And so it is the sequence of control elements and the presence of different activator proteins that elicit different genes to be expressed. I think that's the main idea of this section. So life tends to use the same pieces over and over again, but they just arrange them in different patterns. I hope this helps, and let me know if you have questions.